Artificial Intelligence Dr. www.abcpharma.net/English. User question. What are the neurophysiological mechanisms underlying post-micturition urethral pain? And how do these differ between acute and chronic presentations of the symptom? Claude AI answer. This is an excellent question that delves into the complex neurophysiology of urethral pain. The mechanisms underlying post-micturition urethral pain can indeed differ between acute and chronic presentations. Let's break this down. Neurophysiological mechanisms of post-micturition urethral pain. 1. Nociceptor activation. The urethra is innervated by a delta and C fiber nociceptors. These nociceptors can be activated by mechanical, chemical, or thermal stimuli during or after urination. 2. Peripheral sensitization. Inflammation or injury can lead to sensitization of peripheral nerve endings. This results in a lowered threshold for nociceptor activation. 3. Central sensitization. Prolonged nociceptive input can lead to changes in the central nervous system, particularly in the dorsal horn of the spinal cord. This can result in amplified pain signals and increased pain perception. 4. Neurogenic inflammation. Activation of nociceptors can lead to the release of inflammatory mediators, e.g., substance P, calcitonin gene-related peptide. This creates a feedback loop, further sensitizing nociceptors. 5. Autonomic nervous system involvement. The autonomic nervous system plays a role in bladder and urethral function. Dysregulation can contribute to pain and altered sensation. 6. Pelvic floor muscle tension. Increased tension in the pelvic floor muscles can compress the urethra and contribute to pain. 7. Referred pain. Pain from adjacent structures, e.g., bladder, prostate, can be perceived as urethral pain due to shared neural pathways. Differences between acute and chronic presentations. Acute post-micturition urethral pain. 1. Nociceptor activation. Often due to direct irritation or inflammation of the urethra, e.g., urinary tract infection, chemical irritants. Primarily involves peripheral mechanisms. 2. Inflammatory response. Acute inflammation leads to the release of inflammatory mediators, sensitizing nociceptors. This is often reversible with resolution of the underlying cause. 3. Reflex muscle tension. Acute pain can cause reflexive tensing of pelvic floor muscles, potentially exacerbating symptoms. 4. Limited central nervous system changes. Short-term pain typically does not induce significant central sensitization. 5. Autonomic response. Acute pain may trigger temporary changes in autonomic nervous system function. Chronic post-micturition urethral pain. 1. Central sensitization. Prolonged nociceptive input leads to changes in the central nervous system. This can result in pain amplification and altered pain processing. 2. Neuroplastic changes. Chronic pain can induce structural and functional changes in the brain and spinal cord. This can lead to pain persistence even in the absence of ongoing tissue damage. 3. Altered pain modulation. Dysfunction in descending pain modulatory systems can contribute to ongoing pain. 4. Chronic pelvic floor dysfunction. Persistent muscle tension or weakness can develop, contributing to ongoing symptoms. 5. Autonomic dysregulation. Long-term changes in autonomic nervous system function can occur, affecting urethral and bladder function. 6. Psychosocial factors. Chronic pain can be influenced by psychological factors, e.g., anxiety, depression, and social context. 7. Sensitization to normal stimuli. Previously non-painful stimuli, e.g., normal urine flow, may become perceived as painful. 8. Neurogenic inflammation. Chronic activation of nociceptors can lead to ongoing neurogenic inflammation, creating a self-perpetuating cycle. 9. Altered gene expression. Chronic pain can induce changes in gene expression in both peripheral and central nervous systems. Key differences. Acute pain primarily involves peripheral mechanisms, while chronic pain involves significant central nervous system changes. Chronic pain often involves more complex and widespread changes in the nervous system. The reversibility of neurophysiological changes is generally greater in acute pain compared to chronic pain. Psychosocial factors play a more significant role in chronic pain presentations. Understanding these differences is crucial for developing appropriate treatment strategies. Acute pain management often focuses on addressing the underlying cause and providing symptomatic relief. In contrast, chronic pain management requires a more comprehensive approach, potentially including interventions targeting central sensitization, pelvic floor rehabilitation, and addressing psychosocial factors. Future research directions might include 1. Identifying biomarkers to differentiate between acute and chronic urethral pain mechanisms. 2. Developing targeted therapies for modulating central sensitization in chronic urethral pain. 3. Investigating the role of neuromodulation techniques in managing chronic post-micturition urethral pain. 4. Exploring the potential of personalized medicine approaches based on individual neurophysiological profiles.